In our last video, we discussed how prokaryotes regulate gene expression through the use of operons. In this video, we're going to switch our attention to eukaryotes. We'll briefly look at 5 major ways in which gene expression can be regulated, and then we'll focus on modifications to chromosomes that can occur to regulate gene expression. Now, there are a number of steps in this model here where gene expression can be regulated. So, for example, we talked a little bit with the lac operon as an example how transcription can be controlled. We're going to come back to this idea in our next video. But transcription factors and control elements are the major role players in controlling the actual transcription of a pre-mRNA. That pre-mRNA is then produced into a mature mRNA through RNA splicing or RNA processing. This includes the addition of a poly-A tail and the 5' cap in eukaryotes. So this can be another way to control which form of a gene is going to be expressed. Remember that different introns and exons can be put together, and how they are put together will ultimately decide what mRNA is going to be produced and what protein it codes for. Translation initiation can be a point of regulation, and we'll talk about this one in a little bit more detail later on. But in essence, what you need to know here is that the speed at which translation is initiated, or whether it's initiated at all, can be regulated. And in addition to this, RNA stability can also be regulated. Most notably, we talk about how the poly-A tail, its length, can control mRNA stability. Typically, the longer a poly-A tail is, the more stable the mRNA is. And then we also have post-translational modifications as well to proteins that are going to control their expression or control their function. So, for example, insulin is a protein that must, after it's initially made, must be cut and cleaved into a mature form within the Golgi before it's secreted from the cell. And then we've talked in signal transduction pathways about how proteins are, in some cases, always present in those signal transduction pathways, but they're only turned on when they're modified with the addition of something like a phosphate group. Now, the focus of today's conversation is going to be something that occurs before all of this, and that is DNA modification. In particular, we're going to talk about 2 modifications to DNA: histone modification and methylation. So let's go ahead and talk about these 2 modifications that can occur to the structure of the DNA and lead to changes in gene expression. First, what we're looking at here is what's known as a nucleosome. A nucleosome contains the DNA wrapped almost a couple of times around 8 proteins that are known as histones. And these histones we often find will have tails on them, as we're drawing here. Each of these tails are made up of amino acids, and they serve as attachment sites for different modifications. Now, these tails on the histones can be modified. So, for example, what we're showing here are additions of what we call acetyl groups. Acetyl groups are negatively charged, and you may remember that the sugar phosphate backbone of DNA is also negatively charged because of those phosphates that we find there. And so the interaction between these acetyl groups on the histones and the negative charge of the DNA backbone actually cause the 2 of them to repel from each other. This causes the DNA to be loosened from the histones. The loosening of DNA will actually result in this DNA being accessible for RNA polymerase and transcription factors. Therefore, it's accessible for transcription. Removal of these acetyl groups will take the histones back to a more neutral charge. This is going to increase its interaction with DNA, tightening that coiling of the DNA, and thus repress the expression of genes. Now, acetylation and deacetylation of these histone tails are only one of the modifications that can happen to them. And actually, current research is trying to understand what we call the histone code. In other words, what combinations of modifications lead to the active chromatin state or the repressed chromatin state of DNA. In this repressed chromatin state, shown over on the left-hand side here, DNA is tightly coiled around those histones, and DNA is not actively being transcribed in these cases. However, over on the right-hand side, where we have active chromatin state, this would represent something like the acetylation of these histone tails. What we find is that DNA is more loosely coiled, and that's going to allow for those transcription factors and RNA polymerase to bind to the DNA and activate transcription. Now, another form of regulation of DNA is known as DNA methylation. You may recall that a methyl group is a CH3 group that has no charge and is nonpolar. And in fact, the addition of these methyl groups to DNA is called methylation, and it increases the binding of the DNA to histones. And so in regions where we see methylation, these genes are often silenced or turned off, whereas those without methylation are more likely to be active. Now, there is evidence to suggest that these methylation patterns are inheritable from one cell to the next, and this is a growing field of research known as epigenetic inheritance. For example, children who were in utero during the Dutch famine from 1944 to 1945 have different methylation patterns that they have carried out throughout their history, and often are less have a lower mass or less weight than their counterparts that were born before the famine or after the famine. In review, this video shows us a number of steps in which gene expression may be regulated. Today, in this particular video, we focused on DNA modification. We'll talk in the next video specifically about transcription control.